So why don't you guys maybe take a moment to introduce yourselves and your companies, say a little bit.
So I'm Herpin, I'm the CBO of Caribou Biosciences. For those who don't know Caribou, so we're a gene editing company started with CRISPR technology coming out of Jennifer Doudna's lab at Berkeley. We developed now a second-generation gene editing technology that we call Chardonnay, and we've been using that to develop our own therapies in the areas of CAR T cells, and we try to make them allogenic. By background, I've been at Caribou for about a year now. Before that, I spent quite a bit of time doing business development in large pharma. I was at AstraZeneca for 7 years and at BMS for 11 years before that. Originally, I studied as a scientist. I'm a Ph.D. chemist by training.
Yeah, my name is Nell. I'm heading corporate strategy and business development at Casebia. We are kind of similar to Caribou. We are also invested in CRISPR-Cas, but with only a therapeutic focus, trying to create cures and curative approaches in various disease areas. And I came from Bayer, actually, and if you might know, Casebia is a joint venture between Bayer and CRISPR Therapeutics. And the idea, and maybe that's also part of our BD discussion, why I would explain maybe how it came to Casebia, was that at that time in 2014-15, when we were thinking about doing something at Bayer in gene editing, it was still an extremely high-risk area, right? So projects could easily fail. And the idea was really to create an independent entity that is not part of the daily business or the portfolio review meetings, as you know. And as soon as something would happen within Bayer, like financial constraints or whatever, sure, these projects would die first, right? And that's why it was decided to do something outside. And we thought about the joint venture structure as a very smart idea, bringing in a partner that already has knowledge in that field with CRISPR Therapeutics. And that's where we get the IP. And then the funding came from Bayer. Decent funding to really work for 5 years, exploring the technology and trying to bring assets to the patients to get to IND stage.
So in thinking about partnering in gene editing, I think, as you mentioned, IP access is a key component of that. And you both work for CRISPR companies, but we know that, or CRISPR therapy companies, but we know that there are other gene editing technologies, not just CRISPR, but a sort of unifying theme is the IP access. And so partnering is a necessary component of that. And so could you comment a little bit, do you think, some thoughts on how people can access IP? Or whether it should be more accessible or could be?
Yeah, so maybe I can start. So as you mentioned, there's quite a lot of people out there that are interested to get access to some form of gene editing IP. And CRISPR-based IP is just one of the technologies that they can get access to. From our perspective, the way we view these discussions really fall into two buckets. So there's the discussions with the larger players, the larger companies, and those ones can take a variety of flavors. There's people who are interested to get access to gene editing to do something that we're absolutely not doing. So we tend to be focused on immuno-oncology. People might be interested to access gene editing for completely different diseases. And there, it's a fairly easy discussion. And usually, what we're trying to get out of these discussions is resources to progress our own pipeline. Then you have discussions with big players that want to do things that are similar to what we're doing. And then these discussions are a bit more complex, because typically we try to find a way where we can work with them to enable them, but retain the ability to develop our own programs. And then, as our program progresses, there's discussions around our programs themselves. The other bucket of discussions we have are with smaller companies. So there's a lot of biotech companies that, for what they're trying to do, they also need access to gene editing. And there the discussion is usually quite different. With smaller companies, we tend to have discussions that are more around cross-exchanging technologies. So from our perspective, as we try to develop our product, we need access to a lot of additional technologies. These cell therapies, between the CAR, between switches, between additional engineering technologies, they require quite a bit of technology. And so some of the better discussions we have with other biotechs are really in the area where they can bring us something that's helpful for our products, we can bring them something that's helpful for their product, and that usually leads to a pretty balanced discussion.
A true partnership.
A true partnership.
Yeah, for us it is slightly different because due to the joint venture structure, our parent companies have an opt-in right at IND right now. So that's the initial concept, right? I mean, they bring in the value, the IP and the money, so there needed to be some value as well for them. However, I think in general, I mean, there's discussion about this patent pool and all that, right, on CRISPR. I mean, we talked about 7,000 genetic diseases. I mean, the few existing companies, 4 or 5 gene editing companies, will never address all of these diseases, right? On the other hand, I think to get started and to get investments, you have to be exclusive for some time, right? I mean, you have to get the thing started, and you're taking a huge risk. I mean, you have employees, right, that could lose their jobs and everything. So you take the risk, you invest first, but I think in the future it will change, and I think the broad concept is very similar to that, right, where they said, I mean, yes, we get the IP for the therapeutic use to editors, but at some point there will be discussions. I mean, if they don't work on these projects, then it might be available for other companies, right? But I think this initial period, and we've seen this in other technologies as well, right? I mean, we've seen now Alnylam, after how many years, finally they succeed, right? If that would have been spread to many companies, the money would have not been there to invest, right? So I think that's where it will go. And cross-licenses, yes, we get this asked as well, for sure. It's not always easy because you also have to see the value, right? I mean, if somebody would come today and say, I have an in vivo delivery technology that we have tested in NHP, it's working, this is the data, we might talk about that, right? But if they just have like preliminary in vitro data, I mean, that's a different value than what we have already established in the CRISPR field, right? So we have to talk then how to balance that. I mean, you cannot just give away all the IP on that, right?
Yeah, absolutely. But I think it is, you know, you both have mentioned good points about how, you know, these products are going to be very sophisticated and multiple technologies may be needed in order to really accomplish the goals. You need the editing, you may need switches, you need delivery. And so when BD people hear this, they hear license, 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 which of course means royalty, 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 right? So royalty stacking, as we all know, it's the thing we don't like to talk about, nobody mentions, you sort of say it under your breath. I just said it out loud. So, you know, this is a real challenge in the industry, and we know there's a lot of pressure on pricing of the drugs, and so the more of these royalty stackings, it's you know, going to become a bigger issue, I think, as these products continue to progress. So what do you see as some potential opportunities or mitigations to royalty stacking?
Yeah, so in my mind, you know, this is one example where, you know, the discussions when we have it biotech to biotech is very different than when you have it, you know, with another player. You know, if you're having it with a company that's trying to do something similar than you're trying to do, that is facing the same issue of royalty stacking, it's a lot easier to say, well, okay, so, you know, we're both cross-licensing, we're both providing something to the other. Let's try to both be reasonable and actually keep our royalty reasonably low. I think as soon as we start to have discussions with other companies where really you're trying to access something in exchange for money, I think the discussion around the royalty becomes a lot more difficult. Although to be fair, you know, I think as the field is progressing and as, you know, everybody starts to understand that, you know, your royalties are going to come from, you know, whatever target you have, whatever editing technology you have, whatever additional technology you have in your cells, whatever you have to pay on the manufacturing side. I think the whole field is becoming a bit more realistic and a bit more reasonable with respect to ability to offset other royalties.
Yeah, very good point. And I would like to add, so not to view this isolated as royalties, but to bring in milestones potentially, right? So there's a lot of misperception, at least in my eyes, about the value of these approaches, right? I mean, we're talking about cures, right? Hemophilia was mentioned earlier, and maybe that's a very good example. Right now, with the chronic use of recombinant factors, yes, the annual treatment cost is like $250,000 to $400,000 in US, but a cure will not be the equivalent of 20 years of that 400,000, right? So once a patient is cured, hopefully forever with one treatment course, that means this patient will not be on the market anymore, right? So now we're talking different sales here for these products, right? And very often when we do the first, you know, discussions with companies and they say, oh, hemophilia, that's an $8 billion market. Yeah, but that's the chronic market, right? That's not the cure market. The cure market will be much, much smaller, right? We have to admit that. And so you could introduce milestones that are sales-based, right? I mean, if you create a really successful product, then you want to share that, right? And if a lot of patients will be cured, you want to share that benefit, right? And so you could have sales milestones reaching I don't know, like 200 million sales, right? You have another milestone, which in the end is royalties, but you don't have it for granted, right? And with that, you can keep maybe the royalties at a level that makes sense for these early stage projects that we're talking right now, right? I mean, the earliest gene editing project just entered the clinic, so if you want to license that to big pharma, you have to.
Yeah, it's a way of sharing the success,
Yeah.
an alternate approach. Are there any comments or questions from the audience about what we're talking about, IP access or royalties? Anything? No? Okay. All right. So another thing that we talked about in the preparation for this panel is what to partner. Is it best to partner the platform, or is it best to partner the product? And this is not an easy question to answer, and it's maybe not a one-size-fits-all. But when we spoke about this earlier, we talked about the timing, and that perhaps the point in the development of which the partners are discussing may be a key into whether or not it's a platform or a product play.
Yeah, so I think, if you start by looking back a little bit around the deal making that's been happening in the gene editing space with, the big players in that field so far, a lot of the early deals have been fairly broad platform access, covering, 3, 4, 10 products, and usually combined with, one product that's reasonably well advanced to sort of anchor the deal. And I think these deals are actually quite attractive because you combine both platform access and product, you can actually construct something that's quite meaningful and that's quite visible and attractive, and can allow the company to actually progress its own products. The way we've been thinking about it is we're open to partnering products or partnering platform. We actually think that because there's so much need of gene editing for various things, the platform is very well suited to do various types of deal. And as I mentioned, depending on how far the partner wants to play compared to where we're playing, these deals can take various structures. At the same time, as our pipeline develops, we're also very happy to consider particular deals around assets.
Yeah, I would open this up maybe to in and out licensing, right? So for in licensing, we clearly look at platforms, right? Delivery platforms. So we are very much engaged in vivo gene editing. So we're looking for delivery platforms, and there we want to have a real collaboration with the partner, right? Not a single asset kind of like focused deal, but really be able to develop the delivery method in that case further, right? And then really get engaged and have it a dual partnership. For out licensing, I mean, we're not at this stage yet. I see both models, right? And I see disadvantages also with the multiple target deals, because then you're kind of committed to that partner and they usually have a selection possibility, right, which is then limiting your ability to maximize the benefit of the technology. Right. On the other hand, if it's only one, then you have, again, this usual problem with big pharma that this is only one program, and there might be a lack of commitment in case of crisis or whatever, right? I mean, they might immediately say, no, let's, stop this, right? If there's a commitment to a real range of this gene editing is a new technology within our big pharma company, right? Then this is much better, right? So it's pros and cons. I think, both can be done. But for us, most likely it will be more like asset licensing initially, out licensing. As I mentioned, we're working on hemophilia, so that's a kind of standalone indication that you could use, right? But you can build others on, and then you can build up a relationship and maybe work on additional indications with that company. Yeah,
that makes sense. Are there any comments from the audience? Oh, yep. Is there a microphone?
No one was expecting comments. It doesn't even work. Okay, perfect. Well, since I like to provoke people in order to have interesting conversation, there are two examples in the industry of IPs that no one cares unless they really have to. And everyone is talking about allogenic in the CAR-T world, but no one has in mind the fact that there is an IP pretty strong around removing the TCR but no one cares, or very few. Another example is there are people very happy about using IL-7, IL-15 in their cocktail, and there is a very strong IP on that, but no one really cares. So everyone is developing, getting very close to commercial, and then where the IP really become a challenge. So I'm wondering, if I'm starting a company today, and roughly will take me 10 years to get to commercial, based on what I'm hearing, should be better approach not to partner at all and just wait, because, I mean, it's a tough bet, tough risk, but maybe in 10 years, you might not be in the market anymore, and I have free access to your IP anyway. Would that be a strategy?
So you're saying...
Because the IP, from a research perspective, which means up to the end of phase 3, I can play with their IP as much as I want. I'm not getting any money out of it. I'm not infringing anything. I'm doing research, clinical research, but research. Could that be a strategy?
Yeah, so I think what you're saying is even more broadly, you know, as you think about these therapies that are incredibly complicated and potentially that you know, as you try to develop, there's many licenses that you need to get. You know, should you get everything at the beginning before you get started, or should you get everything just before you get to market? Is there things you should not worry about? I think, you know, I think you bring up a very valid point that, you know, even as you progress your therapy, you realize that, you know, sometimes there's things that you might want to use where, you know, there's other people that have IP. And I think, at least from our perspective, it's always you try to see what you can get when you can get it. So, you know, depending on what you need at any one time, there is time when, you know, certain IP is fairly readily available for fairly, you know, well-defined terms. And there's always the question, you know, do you need exclusive or do you need non-exclusive access? And I think, you know, we tend to think about those things all through the progression of the program. Yeah, I think that's the general approach we take.
Yeah, I mean, I would agree to that, and I mean, the level of the project is, or the status of the project is also important, right? I mean, once you have fixed your candidate and you're in the clinic, you cannot change, right? But maybe there's new technologies coming up, better technologies, like second generation, right? I mean, different technologies. So initially, I would not fix it necessarily, but once you have a candidate, I would try at least to explore if you can get it. But it could not be dramatic. But then you also need to consider if you want to partner with big pharma, usually they do not engage in projects where they don't have FTO, right? So it might hinder your partnering abilities if you don't have access to the IP yet.
Yeah, and it's similar with investors. Yeah. You know, as you go and raise funding, you know, the stronger your IP situation, you know, the easier is the discussion.
Yeah. Also, I think, you know, if you're referring to the fact that you can access many CRISPR-based tools, research grade, you know, you may actually get to a point where you're boxed in the corner where you really only can, you know, based on the tool you've been using, there's really only one partner you could go to for the IP, and now you're close to commercial. You're going to kind of be over a barrel there, and maybe not get as good of a deal as you would have earlier. And also, I think another benefit of partnering early is not just the access, but usually it's the partnership, the co-development, the expertise, the know-how that you have access to, which can help you be more successful with the product, and another justification for partnering earlier. Good question.
Thank you.
Any other questions or comments? So another topic that we had talked about, in which you mentioned just before, I think both of you did actually, is partnering with tools and service providers. Because we know that the CRISPR and other gene editing tools work great when they're in the nucleus of the cell, but they have to get there. So there are a couple different modalities that people can use. But what, you know, can you comment maybe on your thoughts or some experiences you've had with evaluating tools and services for delivering gene editing tools into the cells?
Yeah, so I mean, we talked to multiple companies for sure. I mean, there's a variety out there that usually came from other technologies, right? And so the quest will then be to select the one or ones that are most to what you want to use. So we're talking about a different payload, right? I mean, if you think about, we're using mRNA expression of Cas9, for example. So there's some mRNA companies, right? And now you will look, I mean, those were vaccines and other approaches, right? But, so the technology is similar, but we have a different payload. I mean, Cas9 is rather big, right? It's a big protein. And so we look at those, and then at some point you have to select a partner, right? And work on that, moving the technology further, right? I mean, I think there's nobody there yet who has the final product available for licensing or so, right? So we have all the gene editing companies that I know at least of working together with partners, trying to further develop these technologies, right? Same for in vivo delivery, being at AAV. I mean, there's a lot of AAV knowledge, but now we have multiple components, right? We're not talking about a DNA only, like in the traditional gene therapy case, but we also have to deliver the guide RNA, we have to deliver the Cas or the nuclease in general. So that needs to be discussed as well, right? I mean, that has an impact on, suddenly you're talking about manufacturing as well. I mean, we're maybe not touching that too much, but if you need multiple AAVs, think about the Sangamo approaches. There's 3 AAVs, right? I mean, that's not cheap, right? Now you have to coordinate all this, and all these thoughts get into where do you partner? I mean, what is the best? I mean, we are working with Stride Bio on AAV optimization, for example. So we're trying to even evolve the AAVs, right? That we're using, not just using plain AAV, 8, for example, but we're trying to go further, right? And so all these partnerships we're trying to set up, and moving the whole field. I mean, not only the Cas CRISPR field, but also all the technologies that we require.
The associated technologies, other viruses as well.
Yeah. In our case, so we really focus mainly on ex vivo editing, and we typically use electroporation to get the guide and the nuclease. And then we typically use an AAV vector to get the donor DNA when we do a knock-in. So, you know, typically we're talking with companies that have various versions of technologies that can broadly help things get into cells. I think when we have those discussions, there are a couple of things that are helpful, you know, to the extent that people can come to us with the data using CRISPR reagents, that's very helpful. And then the other thing that's also helpful is, you know, having the ability to work with people under MTA, so that at least, you know, we can get our hands on the technology, or we can actually see some of the data with our own reagents. That also makes a big difference.
So what is some data that you would like to see from a delivery tool that would give you some comfort that it would actually work for CRISPR-based tools?
Yeah, so at the end of the day, you know, the data that interests us the most is the data with our own reagents, right? You know, there's particular editings we're trying to do in T cells. You know, the ultimate data that we want to see is how does their technology compares to the way we're currently doing things. The next best thing is to have, you know, something that's reasonably well validated in the field. So, you know, if you look, you know, there's multiple papers published where people have played with technologies to get CRISPR reagents and DNA donor into cells. You know, to the extent that people have comparative data using similar genes, that's helpful to us. Because some of the times we've actually already played with those things in our lab. So, you know, we know what's reported in the paper is doing in our hands, and so having that as a comparison is actually quite helpful.
Yeah, I mean, you talked about ex vivo. We are working in ex vivo as well, but I think that's very similar what we need there. For in vivo delivery, I would say, I mean, in a dream world, we would love to see a large animal species like NHP for sure, right, delivery, and biodistribution data in some sort of animal to see if you're really hitting the right organ, for example, right? If you're talking liver, I mean, the best part would be to just go into the liver, right, which is maybe the easiest for AAV, but then we're talking about other diseases, lung and so on, where you need to be very organ specific, and you have to show that as well, right? But safety data is helpful. That can be, I mean, mouse or rat, right? I mean, in that case, but ultimately, before we would make a decision for sure for any candidate, we would go into NHP, and that's I mean, if you have that already, super, but we understand it's not the cheapest to do, right? So that could be then done in partnership if we see confidence that we would go with such technology. Right?
Yeah. Yeah. Any thoughts from the audience or comments? There's a microphone.
Thank you. When you're considering ex vivo non-viral platforms, do you look beyond the basic efficiency, viability, and yield of the system towards, for instance, the functionality of the cells which result from that engineering?
The short answer is yes, we do. Usually it's sort of the second step. The first step is really we'll look at, the efficiency and how that compares. And then the next step is we try to understand how does that affect the cells and broadly how it fits, with our current manufacturing process. Basically, can we implement it? And at the end, are we getting a better yield of cells?
Thank you.
Yeah. Okay. All right, thank you. All right, and then I think another important consideration in talking about partnering is always thinking about pharma. Because that's, as was mentioned in the earlier panel, not everybody's going to be able to have the sales, marketing, distribution channels. And so even if a small company is able to grow and evolve with the product, really, I think at the very least, by the time you get to commercial launch, partnering with pharma is going to make a lot of sense in order for the product to truly get to the patients and be successful. And so when you think about pharma, I guess, what are your thoughts about when is a good time to bring a pharma partner in? Is sooner better? It's not always up to you, right? But if it were, would it be better sooner or later to bring the pharma partner in?
So to me, it absolutely depends on the indication, right? So there are some indications where you need the patient access, and we heard this earlier, right? The relationship with the patient organizations, families, and so on is very important, right? And sometimes small companies can't do it, right? And coming back to hemophilia, which is one of these indications where the patients are attached to the company that they're using the recombinant factor of, right? And so you need this power to recruit patients even for a phase 1, right? It's much more difficult for a very small company. And we've seen that in the partnering as well, right? We've seen Spark partnering with Pfizer, Sangamo with Pfizer, and others. And so I think that's seen there. We are working on IPEX, which is an autoimmune disease, extremely small patient population. We're working with Seattle Children's Research Institute, so we have patient access. I could imagine that we would continue such an indication into the clinic, right? You don't need a big partner behind you for that. But so I think it always varies by indication. And you can also see that, knowing that from my days at Bayer, where we look at, right? For oncology, for example, we would rather look later, right? To see that there's also some data already, phase 2 maybe, right? Yeah. Because you need to convince your own management that you want to invest into this. For some other indications, you would even go earlier because that's where you want to design the phase 1 and so on, right? That's also very important. Is the big pharma company happy with your design of the clinical studies? And maybe that's where you should partner early, at least to get consulting, right, potentially, on these. You can maybe continue performing it, but at least consult with people that have the experience, right? How they would see this, I think, is very important.
Yeah.
Yeah, from our perspective, I think it depends a little bit of what kind of partnership you're trying to put together. I think, if you're trying to work with a pharma who's trying to access gene editing broadly because, they want to get into cell therapies and it's a new organization they want to build on their side, I think, doing it reasonably early is not a bad thing. I think if it's a pure interest in a particular product that you have, and what they're going to be bringing to the table is really, some development capabilities and commercial capabilities, then I think maybe doing it a bit later is not so bad.
Okay. All right. Any final comments from the audience? We're just about out of time, and I know coffee is awaiting us. No? Okay. All right. Well, thank you, everyone. Thank you to our panelists.
Thank you.